So the next question arises, how the initiation of replication takes place. It was explained by Francois Jacob, Sidney Brenner, and Jacques Cusin in 1963. The model they proposed is called as the Replicon model of replication initiation. They defined all of the DNA replicated from a particular origin of replication as a replicon. So, if the whole DNA will be replicated as a result of single origin, the whole DNA will be called a replicon. As the single chromosome found in E. coli cells has only one origin of replication, the entire chromosome is a single replicon. While in case of linear chromosomes, the presence of multiple origins of replication in eukaryotic chromosomes results into multiple replicons, one for each origin of replication. So the replicons are equal to the number of origin of the replication. The replicon model proposed two components that controlled the initiation of replication, and these two components are the replicator and the initiator. The replicator is defined as the C-secting DNA sequences that are sufficient to direct the initiation of DNA replication. So along the DNA molecule there are some specific sequences which starts the replication initiation. This is in contrast to the origin of replication. So this sequence of DNA sequence, this is not origin of replication. Origin of replication is something different, and that is the physical site on the DNA where DNA is unwound and DNA synthesis initiates. So keep in mind that replicator is a sequence along the DNA molecule, but origin of replication is the physical point on the DNA molecule from where the replication initiates. Although the origin of replication is always part of replicator, because the replication will always start from that sequence, that particular sequence, because that particular sequence will direct the initiation of replication. So this origin of replication will always be part of replicator. Sometimes the origin of replication is only a fraction of the DNA sequence required to direct the initiation of replication. So it means it is not necessary that the origin of replication is the whole sequence of replicator, but this origin of replication may be a small part of that replicator sequence. The second component of the replicon model is the initiator protein. This protein specifically recognizes a DNA element in the replicator and activates the initiation of replication. So on a DNA molecule there is a replicator site, there is a specific sequence. So this initiator protein, it identifies this sequence from this replicator. It identified some specific site from where the initiation of replication will take place. All the initiator proteins select the sites that will become origins of replication. Whatever the site selected by initiator, that will become the origin of replication. The initiator protein is the only sequence specific DNA binding protein involved in the initiation of replication. So here in this figure you can see this is a DNA molecule, and here in this molecule there may be some sequence which is replicator. This initiator protein, this will identify a site. For example, it has identified this site, and this point will become the origin of replication, and then the replication forks start growing towards the opposite directions. So in this way, the replicator and the initiator proteins work together to start the replication. All the remaining proteins other than initiator proteins, they required for replication initiation do not bind to a DNA sequence specifically. They may bind to the DNA, but non-specifically.